this doesn't work, so never mind. Remember that, okay? All right. First of all, I want to thank you all for coming tonight. I'm Brian Cummings. I'm actually Associate Dean for Faculty Development in the School of Medicine, a founding member of the Stem Cell Center here at UCI. So I'm old is probably the right way to look at it. Normally, these talks are introduced by Eileen Anderson, our director, but Eileen is actually at a spinal cord conference in Texas and left the task to me to introduce myself and my postdoc, Johnny Hasselmann. So the way these typically work is that a faculty member and their postdoc or graduate student or a clinician and a basic scientist, they team up to give a public lecture. And we welcome interruptions. We are recording this for posterity and to post on our YouTube channel. So if you do ask a question, make sure I can hear you, and I will repeat your question so that everybody can hear what the question was. Tonight's topic is unlocking hope using stem cells to better understand traumatic brain injury. And I'm going to give you a little outline here. And that doesn't work. Super. I'll give you an outline of tonight's seminar. So first, given these trying times and the State of the Disunion address tonight, so actually I thank you all for coming and hearing science instead of politics. But because of my interest in politics, I'm going to dive a little bit into why should we be wasting money on basic science research or not. Then I'll give you some background on traumatic brain injury, and then specifically talk very briefly about a stem cell therapy for acute traumatic brain injury, the kind of motor vehicle accident injury, not the mild concussions that are part 4 of this talk. Then we'll talk about how we model concussions that are frequently happening in athletes or in our military. And then finally, Dr. Hasselmann will come up and talk about a CIRM grant that we have that Johnny mostly wrote. That's why he's going to talk about it, because I don't understand it. And he's going to talk about how we can manipulate and genetically engineer microglia to possibly treat brain injuries in the future. So hang on for the best part, which is at the end. So, as I mentioned at the beginning, though, science and funding for science is under attack. If you're on Twitter or following the news, Musk and Doge in particular are actually picking studies that sound silly and attacking them. The classic one, this one that got millions of hits on Twitter, is that scientists are funded to spray bobcat urine on alcoholic rats. And, this is insane. Or studying the sex life of beetles, and we're coming to take your jobs away. So let's stop wasting our money on that. So I thought I would pick on these examples right here to give you a little bit more background into some of those studies. So maybe not sex in beetles. This is sex in bacteria. How do bacteria replicate without the extreme heat in some hot springs denaturing their enzymes? How could this happen? And so if it was 1967 and you're the director of the National Science Foundation, would you invest 150,000 of the public's money in studying sex and replication in bacteria? Who's going to make that investment? Well, there's one or two scientists in the audience nodding, but I don't know. Well, it turns out that funded study ended up in the discovery of the Thermus aquaticus bacteria that lives in near boiling water in hot springs in Yellowstone and has an enzyme called Taq polymerase that helps it copy its DNA when it replicates. So it turns out that high heat enzyme called Taq polymerase is the foundation of polymerase chain reaction replication. It's how we are able to make billions of copies of DNA, and that this PCR method has been called the Xerox machine of DNA. What's it good for? Medical diagnostics, making vaccines, detecting viruses, solving crimes with DNA evidence. Big Pharma is using it to develop life-saving drugs. Anyone who's done a genealogy test or Ancestry.com, PCR was behind the scenes there. And this has turned into a 10 billion per year industry, 10 billion. So that 150,000 was probably a pretty good investment. Let's look at another example. How do you cut and join DNA from one species into another species of E. coli lowly bacteria? Why should we care? It ends up with recombinant DNA technology and the discovery of how we could make human insulin in bacteria. So this human insulin, now we can produce it genetically modified, it's human, but engineered in huge vats from bacteria that have been genetically engineered, or recombinant DNA technology. This has resulted in millions of Americans having cheap insulin that they can take. It's led to the discovery of hundreds of genetic diseases. And this is a $100 billion a year industry as well, funded by the National Institutes of Health. And the last one is my personal favorite example here. I didn't realize this, but Gila monsters only eat 3 or 4 times a year. What mechanism allows them to do this? Who cares? Does anybody see where this one's going?
Space.
We're not going to space. This Gila monster, although I misspelled it at one point and called it a Glia monster, you will get that joke later in the talk if you pay attention to Johnny. That's not a Glia monster, it's a Gila monster. The discovery of extendin-4 is a hormone in the saliva of the Gila monster. That hormone is analogous to glucagon-like peptide GLP-1, which is the foundation of the modern anti-diabetic drugs, blood sugar, and appetite control. Millions of Americans are benefiting from this discovery in the saliva of the Gila monster. And this industry is already valued in the billions, and it's just started. It's just growing. So I think we can agree now. Of course, I'm the scientist. I cherry pick some good examples. There's lots of bad examples too, but that's the problem. How do we know what's the good and what's the bad? We got to do the experiment and find out. So the other point I'd like to make is that in terms of the federal investment in research and development, we're at the lowest level of federal support for research as a percentage of the GDP, or gross domestic product. We're at the lowest level in 60 years. So why did it peak in the 60s? Well, the answer is down below there. That was the space race, and we invested a ton of money in getting to the moon, putting satellites out there. But that has steadily declined over my lifetime. Yet still, investment in basic science is incredibly fruitful. For every $1 of publicly funded basic science, it generates $8 of additional research expenditures by industry. For every $1 of a clinical research trial, it stimulates $2.50 roughly of further expenditures. So I would just point out, in these politically fraught times, you're in the audience, hopefully you're supporters of science, but if you want to reduce the deficit and stimulate the economy, cutting NIH and cutting NSF is the last thing you'd want to do. You want to raise funding for basic science to generate even more economic activity, not cut it. But that's not what's happening. What's happening is just a random people who don't know science are taking a razor to anything they can get their hands on. All right. End of the is science good or bad? That's your decision to make, and please support us with your tax dollars. Now a bit of background on traumatic brain injury. So traumatic brain injury is really a silent epidemic. It's the #1 killer of children and young adults in America. And it affects almost 2 million Americans each year, and up to 4 million if you include concussions in that figure. And that's more than brain, breast, lung, and prostate cancer combined. TBI is a huge problem. There's almost 5.5 million Americans living with a disability related to TBI, and that's approaching the levels of individuals with Alzheimer's disease, a huge public health problem. Also, traumatic brain injuries are the signature combat of folks coming from more recent combat, Afghanistan, Iraq, Iran. Those wars are more survivable because of the equipment they have, but then they result in individuals with greater, more likely brain injuries and PTSD. And finally, there's been 25 completed phase 3 clinical trials for TBI, and all of them have failed. This is definitely an area ripe for further investigation. So what is a traumatic brain injury? It's simply defined as an alteration in brain function or other evidence of brain pathology caused by an external force. So not an internal injury like a stroke, but an external force, typically a blow to the head or a blast nearby. And we distinguish that from a concussion, and a concussion is a subtype of traumatic brain injury. So a concussion is when a blow to the head causes the brain to move around inside the skull and bounce against the inside and cause minor bruises along the surface and slightly into the tissue of the brain. And we're going to model this later in my talk. So what are the primary causes of traumatic brain injury? Well, the number one is falls, and this is a bimodal distribution. These are falls in children as they're learning to walk, and they're falls in the elderly as they have difficulty walking. So falls are the number one cause of TBI, and then being struck by objects or walking into objects like using your iPhone and walking into a lamp post, or motor vehicle accidents are the next biggest causes of traumatic brain injury. So what happens in the brain? We're going to focus on, there's a lot of things that happen in the brain. We're going to focus on just two things because they're going to become relevant later on. And the first is that traumatic brain injuries are associated with inflammation. So I'm going to use my pointer here. On this upper panel are control patients stained with an antibody to microglia. Johnny's going to talk more about microglia later, so pay attention. These are human brains at autopsy showing that we generally have a low level of microglia. But if we've had a brain injury, then there are more microglia present after that injury. And these microglia remain inflamed. They're the brain's immune cell, and they remain inflamed months to years after a single injury. So it's a profound, prolonged deficit. And then the other thing we're looking at here is white matter atrophy. So this cluster is individuals at autopsy who were uninjured, and what's the size of their corpus callosum, or their band of white matter. And I'll show you rodent corpus callosums in a minute. So this is human white matter, the normal volume, and then if you've acutely had an injury or you've had an injury for a year or later, you have atrophy of your white matter. So that's not a good thing. So now, with that background on TBI, let's talk very briefly about a CIRM-funded grant that I have. It's coming to a close, but it's called a TRAN1, or a Translational Award. It's after an early translation award that I received from CIRM, then a discovery award, then this TRAN award, and if I'm successful, it'll lead to a clinical award. That's the plan. So back in 2010, when I started this work on a cell line, we imported a human embryonic cell line called Chef6 from England, and we devised a method to take Chef6 and grow it. The Chef6 was originally from an animal cell culture media and an animal feeder cell line in the culture. And that's not going to be a good way to manufacture cells for a human clinical trial. So the first thing we did is we tried to adapt a protocol to transition these embryonic cells from animal feeder conditions to animal-free or xeno-free culture conditions, and generate a batch of cells here that are human neural stem cells that are multipotent. So this is the product that we're making, and just for the purpose of tonight's talk, we're calling them HNSCs, or human neural stem cells. That's all you need to know. Now what you need to know is what happens in an animal after you give it an acute traumatic brain injury. So we're looking down on a swimming pool, and you can see this little square there. I'm going to click the mouse here. If we put a rat into this swimming pool and allow it to swim around on day one, it doesn't know where that hidden platform is because the water is milky, cloudy white. So the rat swims around and it tries to learn where is that platform, and it takes it quite some time on day one. But if we look at day 5, this is the same rat being put into the water maze, and it swims right to that platform and it climbs out. So we can track this using heat maps and digital video equipment, and we can get maps of how long did it take and how fast, what's their learning strategy and all that kind of thing. And if we look at that, we can see that animals that are acquiring where the object is, where the platform is, if they're sham animals or they got a stem cell therapy in blue, they learn very well. If we reverse that, that is, we flip where the platform is and move it to somewhere new, that's very hard to learn. The animals that are a sham animal, they do learn this over time, and the stem cell transplant animals learn that as well. But the injured animals that got a vehicle control, they are unable to learn this reversal task. So the stem cell therapy is improving learning and memory. And I didn't show you the data, but it's reducing anxiety in these animals as well, very common symptom of traumatic brain injury. We don't see any tumors in these animals, and they protect the host neurons and actually differentiate and turn into new neurons themselves. And finally, they reduce inflammation. So this CIRM-funded work, what's up next is I need to submit what's called a pre-IND or an investigational new drug application to the FDA. And based on the FDA's feedback, I would then be submitting a clinical grant to CIRM to fund the manufacture of this Chef 6 neural stem cell product here in UCI's Good Manufacturing Practices facility. So we have a newly built facility on campus that is a facility where we can actually manufacture human cell products that the FDA will allow us to put into people. So that's where we're going. And that was 10 years of work, actually, yeah, 10 years of work in 5 minutes. But I do also want to acknowledge my laboratory that this is not work that anyone can do alone. This is a huge team project. It's taken a lot of time over the years and a lot of people involved. In particular, I do want to point out we have a collaboration with Marcel Dottie at the Southwest National Primate Center, and Chef 6 HNSCs are actually in non-human primates right now, and we're collecting that data as our last bit of the IND package. And then I also have a set of clinical collaborators here at UCI, like Michael Lopez is in neurology, as well as Lenny Grossman and Jeff Chen's a neurosurgeon here, who did the neural stem trial for TBI and stroke. So we have on board the expertise to do this clinical trial at UCI, and hopefully we'll get the funding from CIRM to do that. All right, next quick bit here is how do we model concussions in rodents instead of an acute car accident level injury? How do we model concussions, and what does that look like? So why do we want to do this? I don't know how many of you folks have heard of chronic traumatic encephalopathy. That's a mouthful, as you can tell. So we just call it CTE. CTE, what is it? It is the signature injury of boxers or dementia pugilistica of NFL football players. So shown in the middle here is Ann McKee with some human brains donated from NFL football players. And the first NFL football player was actually diagnosed by Bennett Amalu in 2005. This is the brain of a 50-year-old showing Alzheimer's-like pathology, which is actually the hallmark of CTE in a 50-year-old football player from the Pittsburgh Steelers. So with that start, Ann McKee then looking at autopsies of football players has found that nearly every NFL football player who donated their brain to science. Now, obviously, a lot of them don't donate their brain to science, but everyone in her autopsy case except one had CTE upon pathological diagnosis. And as well, most college and pro football players have this pathology as well. So you can see the stages. It gets worse over time. It gets worse the number of hits that you've had. And Ann McKee's actually been able to calculate that for every 2.5 years that you play football, you're doubling your risk of getting CTE. So the other big thing is, and why we're very interested, is that CTE is found in 30% of combat veterans. So veterans are exposed to grenades and mortars and blasts and IEDs, and these things impact the brain, cause concussions, and they come back with symptoms and problems we're hoping to be able to treat. So the idea is, can we model that in an animal? Can we model concussions so that we can actually help people? This affects, as I mentioned, the military. It affects people in martial arts, mixed martial arts, soccer players, hockey players, you name it. Anybody who's getting a concussion as part of their sport is vulnerable to CTE. So what does this look like in the animal? We call it repeat mild closed head injury. So this is RMCHI, and we can model this by taking an animal and putting them on a foam pad. They're anesthetized, so they don't feel it, but that piston you see above their head comes down and impacts their head, and their head bounces. Then we wake them up, and what we can do is we can do that once, or a one-hit model. We can do it 5 hits every other day, 10 hits every day for 10 days. And then we can also anesthetize as a control sham animals that don't get the hit from the piston. They're just anesthetized 5 or 10 times in a row. And then we can ask, what does that do to animals? And if we look at this graph here very quickly, basically what you're seeing is this is a motor learning on a rod that's rotating. How long can the animal stay on that rod before falling off? And what we see is if they've had 10 hits, they're visibly impaired on that task. But even with 10 hits, they recover by 10 days. So there's a short deficit with 10 hits. There's no deficit with 5 hits or 1 hit. So this is a very mild concussion, but um, there there are repercussions. So even though their motor recovery and motor learning is a return to normal in 10 days, their emotional and anxiety levels are still high. And in fact, they're permanently altered 1, 2, and 6 months after getting 5 hits in this model. This is a time out in the open in elevated maze. So uh, rats and mice are prey animals and they don't want to be caught by a predator, so they hide near the walls and they shouldn't be going out into the open. So how much time they spend out in the open is a sign that they're anxious and that they've lost their fear of open spaces. Not a good thing. I also showed you the water maze previously. This is just demonstrating that learning and memory in the water maze is impaired if we give these animals repeat mild concussions or repeat mild closed head injuries. So what does it look like in their tissue? So I showed you a graph of the human corpus callosum shrinking years after a TBI. This portion in the middle here, stained in brown, the brown is a stain from myelin basic protein, or what wraps the axons in your white matter. And it's a nice thick corpus callosum in the center of the brain here. You're looking at just the right hemisphere, the left hemisphere is cut off. So that is how thick a brain should be. In the one-hit animals, there's a little bit of decrease in the staining, but no decrease in the thickness of the corpus callosum. But you can see very obviously that at 5 hits and 10 hits, there's a great loss of the white matter in the corpus callosum. And we can actually build 3D reconstructions of that. And I think you can appreciate that the sham animal at the top, their corpus callosum is much thicker than the 5-hit animal down at the bottom, where there's thinning of the white matter. So this model, not only are there behavioral and emotional deficits in this model, but there's pathological changes that mirror what we see in people. And then the last thing I'll mention before I turn it over to Johnny is that if you look in the corpus callosum, that same area, now we see these microglia, the brain's immune cell, filling that area where the white matter is damaged and responding to that injury. And these are the same microglia. These are rodent microglia, but the same microglia that Johnny's going to talk about next. So with that, I'm going to turn it over to Dr. Hasselmann.
All right. Thank you, Brian. Excuse me. So, as Brian mentioned, my name is Johnny Hasselmann. I'm a postdoc here doing work with Brian in his lab on TBI. We have a grant together studying how these cells, microglia, respond to brain injuries and trying to figure out if there's something we can do with engineering these cells to better respond to these injuries and improve long-term outcomes in people. And so first and foremost, kind of what are microglia? Brian sort of mentioned it, that your brain's immune cells. The reason that's important is because probably heard of the blood-brain barrier, separates the immune system that takes care of the rest of your body from your brain. That system can't get in, so it needs its own immune system. And so you have microglia in there, which essentially respond to anything that goes wrong in your brain. And this is what they look like. So they have these little bodies and all these arms stretching out, kind of like an octopus. And this is what they look like when they're functioning in the brain. So this is actually a mouse. We thinned its skull. We put a microscope right up against the skull and took a video of those microglia in the brain. And as you can see, those arms are constantly moving, looking, checking the brain, surveilling, seeing if anything is wrong. They do that 24/7, 365, constantly making sure that your brain is healthy and functioning properly. But what does that really have to do with brain injury? Well, in this experiment, we're doing the same thing here. Thinned out skull, microscope on the brain. We shot a laser in where this red box is to create a little tiny brain injury. And this is what these microglia did. See the time frame over here. It's not a matter of minutes. So within minutes of that injury, they start racing all those processes in. They start checking out that injury, figuring out what do we need to do here? What is this problem? Can we stop it from spreading? Do we need to clean it up? Do we need to call for help? What do we need to do here? They start trying to figure out what they need to do to respond to that injury. And so in this sense, these microglia are the first responders of your brain. They're your first line of defense when something goes wrong. And that's really what makes them interesting, right? That's why we want to study them. And normally we do a lot of this work in mice. There's a bit of a problem, and that mouse microglia aren't identical to human microglia. I want to help all of you. I want to help humans. And so I need to know, how do human microglia respond to human brain injuries? And the more we've learned about mouse microglia, the more we've realized that there's actually some pretty serious disconnects between the two. And so what this graph is showing you here is just a selection of genes. But in green are genes that are really, really similar between mouse and humans. Some of them are nearly identical. So like if you wanted to study, say, this cat8 gene here, if you studied in a mouse or human, probably wouldn't matter. They're essentially identical genes. However, as we move down and we get into some of these genes that are blue, those are really dissimilar between mouse and humans. If you get into these gray genes, these are a couple of genes that mice don't even have. You want to study these, you can't study them in a mouse. You have to study them in a human. And like I said, this is just a subset, but we have 20,000 genes. And a bunch of those are really important for microglia function. And a lot of those are very different between the two species. And so we kind of ran into an issue where if we want to understand human microglia and human disease, we need a way to study actual human microglia. Most of you don't want me going into your brains and looking at your microglia, so we needed a better way to do that in a lab. And fortunately, right here on campus, we do that. We're in the Stem Cell Research Center, which suggests that we're using stem cells somehow to do that, right? And the idea there is that stem cells can become anything that they want. They can be any cell type. And so the Blurt and Jones lab right here in the Stem Cell Center pioneered a way to take human stem cells and make human microglia from them. And this is a graduate student. This is where I did my PhD work. A graduate student just before I joined the lab figured out basically looking at development in humans, where you go from embryonic stem cells as the cells or as the human body develops. There's a whole bunch of signals that tell those stem cells to become HPCs into EMPs. Eventually, these EMPs go into the developing spinal cord and brain. There's a bunch of signals in that developing brain that then tell those cells to become microglia. And so the student, Edsel Aboud, decided, well, we should just be able to do that in a dish. If we take stem cells in a dish and we give them those same signals, we should be able to make microglia. And he did. And this was really pioneering at the time. But basically, he took stem cells in a dish and he added some of these yolk sac proteins that occur during development, and they became these HPCs and EMPs. And then he added some of these proteins that are present in the developing brain, and those cells became microglia, human microglia in a dish that we could keep in an incubator. And that was super exciting. So all of a sudden, we had millions of human cells that we could study in the lab. And then a lab at UCSD said, microglia in a dish, they don't look like microglia in your brain. And so what they actually did, they took some human brain samples. People donated their brains after they died. And they took some of those microglia out and they studied them right away. And they took a subset of those and they put them in a dish in an incubator for 7 days and then looked at them. And it turned out they look completely differently. So 7 days later, those microglia that are in a dish look nothing like the microglia from the brain anymore, which now raises a problem. I have millions of microglia in a dish, but I want to understand how to study microglia in your brains to help people with brain injuries. And so we needed a way to fix that disconnect. And so when I came in and started my PhD work, the idea was, well, that's simple. Let's just put human microglia in a mouse brain, right? Simple solution. Totally makes sense. And what we did is basically the mouse brain, when it's first born, 1 or 2 days after it's born, their brain is still developing. And the idea was, well, if we take some of these microglia and we put it into the developing brain, just like during human development, those cells should spread throughout the mouse brain and we'll have human microglia in a mouse brain. And luckily, for the sake of my PhD work, it worked. And so what we're doing here, this is something called immunohistochemistry or IHC. There's a picture of a mouse brain that we took on a microscope. We're looking in from the side. The eyes would be up here, back of the head is here. Everything you see in green is a human microglia living inside of a mouse brain. And so the fact that this worked now solved a bunch of these problems. We're now able to take human microglia. We don't need to have them in a dish. We can put them into a brain environment where they should function normally, which then brought us to the point of we have these great models in Dr. Cummings' lab where we can study traumatic brain injury. So we were ready to put all of those things together,
Wait a
right?
minute. How did you get green cells?
Oh, yes, that's a good idea. Sorry, I went through, I'm passing it again. Oh no. Technology. I'm a scientist, I swear. Yeah, and so again, these cells actually had a protein in them called green fluorescent protein, which tying back to what Dr. Cummings talked about earlier, these days this work probably wouldn't get studied because that protein comes from a jellyfish. And so this was some random grant that was funded at some point to study these proteins in jellyfish. They're actually now used across almost all of science. We use these fluorescent proteins to label things, label cells, label proteins. And it really has revolutionized science, Nobel Prizes, everything. And so sometimes these weird projects 10, 20, 30, 40 years later are actually critically important for the work that we do, and they will continue to be for the foreseeable future. Good question. But anyway, so kind of the point we're at right now, we know we have this traumatic brain injury model in these mice, And it leads to these behavioral changes that are very similar to things that human beings experience when they're injured. We know that there's structural changes to the white matter, to the cortex in the brain. We know that microglia rapidly respond to these injuries. We have a model where we can study human microglia in the brain. And so we figured let's put all these things together and let's learn a bit more about how microglia respond to brain injuries. And the way we did that, you could probably guess, we took some human microglia and put them in a mouse brain, got a whole bunch of human microglia in the brain, aged these mice to about 2 months. That's the start of adulthood for mice. And so 2 months is kind of where a lot of experiments start. Anesthetized them, put them on this device with that pneumatic piston that you saw a little bit earlier, and we gave them 5 hits. And that's spread out over 10 days. So they get hit, and then they have a day off, and they get hit. And that's reminiscent of, say, a football player. They're not going to get hit just once. They're going to go in, they're going to get hit a bunch of times. A boxer, an MMA fighter, they're not just getting hit once, right? They're going in there having a fight, they're getting hit a bunch of times. And so that's why we want to model this with multiple hits. So it's more reminiscent of what these people are experiencing. They're not hard hits, they're these mild, repetitive hits. And that's what leads to this disease like CTE. And this is kind of the core of that grant that you mentioned. So we did these hits, and then we waited 24 hours after injury, 10 days after injury, 6 weeks, 12 weeks. And at each one of those points, we pulled these microglia back out of the brain and asked how are they responding to these injuries. And we're doing this in a couple ways. One of the ways is IHC, right? This picture that you saw up here. That's taking the brains and actually putting them on a microscope with these fluorescent proteins and physically looking at the microglia. The other thing we can measure is something called RNA, which we're going to touch on next. And we measure RNA to kind of try and figure out what jobs we think the microglia are doing in a brain. And every time I show this data, I tell a story. This is like a year's worth of work, cutting edge data, super excited about it. And I have an 8-year-old daughter. And I showed her this when I got the data because I was excited. And this is the data. And she looked at me and said, that looks like a preschooler's piece of artwork. Which kind of broke my heart. Like, it's not. It's really important scientific data. She's like, no, it's colorful dots on screen, Dad. Um, and so I don't want all of you thinking that. So we're going to walk through this data here, and then hopefully you'll believe me that it's actually important data and not just cheap artwork. But what this is this is using a technique called single cell RNA sequencing. And so as that sounds, each one of these single dots is a microglia. It's a single cell that used to be inside of a brain. And the colored groups are groups of jobs. So we measured the RNA, and if 2 microglia had similar amounts of RNA, they got lumped together into the same color. If they had really different levels of RNA, they got put into different colored groups. And so these groups sort of define clusters of microglia that are doing similar jobs in the brain. We don't know what those jobs are yet, but we know that they're kind of grouped together doing similar jobs. Okay. And so we're measuring RNA to figure out those jobs. And give a little background on what RNA is so this makes sense is you've all heard about DNA, right? It's the instructions to make you. In a lot of ways, it's like a cookbook. It's a book that has all the recipes for how to make you. The genes in your DNA are like pages in that cookbook. RNA are the instructions on those pages. So those recipes are your RNA. And they're recipes for making proteins. Proteins are the tools that cells use to do work. And so RNA is the recipe for how to make a protein, which then allows a cell to do work. And so by studying what RNA are being used in a cell, we can make a guess about what proteins are doing and what jobs they're doing. And so what this plot is showing over here is all of the microglia taken out of a brain. And this is a lot like walking into, say, a restaurant kitchen. And there might be 20 cooks in there, but all those cooks are using slightly different tools and slightly different recipes. Oops. There. So one of them might have a frying pan and be over the stove. One of them might be chopping up lettuce, making salads. One of them might have a blowtorch, making creme brulees. One of them might be manning the deep fryer. Sometimes people use microwaves in kitchens. Uh, they're all still chefs. They're all still cooking, but they're using slightly different tools and slightly different recipes to do slightly different jobs. And that's like your microglia. And that's what this data is showing us. So you have all of these microglia in your brain using slightly different RNAs and slightly different proteins to do slightly different jobs, but working together to keep the brain healthy. So that's what these colorful preschool-esque dots are telling us. Why I was so excited about it. And so diving into the actual data, we have a little over 32,000 microglia here. And this plot contains microglia that were taken from all of these time points. They're all grouped together. So 24 hours after injury, 10 days, 6 weeks, 12 weeks. So once they're all analyzed and put together, we can then split them back out to figure out what differences are occurring. We also have sham animals, so animals that were healthy and didn't get hit. They just got that anesthesia, woke up with no impact. And we have animals that did get hit 5 times, 24 hours later right now. And what we notice right away when we compare these is that this green cluster, whatever this green job is, seems to get a lot bigger. When you hit mice in the head 5 times, 24 hours later, there's a bunch more microglia doing this green job. And if you go and count them, there's actually about 60% more cells doing that job. So that seems like a pretty important job. That seems like maybe like that video I showed you, those cells that rust right into that injury minutes afterwards, they're probably doing this green job. Those are probably those injury responsive microglia. And if we look at 10 days later, that actually starts to come back down. There's only about a 20-25% increase. By 6 weeks to 12 weeks later, it goes right back down to baseline. So what this tells us is that you get hit in the head, and within 24 hours to 10 days, a bunch of microglia respond to the injury, and then over time, they go back down and seemingly go back to normal-esque. There's still other jobs. We're just focusing on the green cluster for now. And so we think that's that early injury response, right? There's a little bit of a problem. We're excited. We think we found it. We saw this thing in the video. A few years later, we came back. We found what we think are microglia doing that job. But as I mentioned, we're measuring RNA here. If you walk into a kitchen and there's recipes for creme brulee spread all over the kitchen, maybe they made creme brulee, but it doesn't actually mean they made creme brulee. Maybe they were getting ready to make it tomorrow. Maybe they were just reviewing the recipe. So it doesn't actually mean that they did the job. And so this is a prediction of what we think is happening in the brain. But to prove if this prediction is reliable, we need to measure the protein. And just like I showed you earlier, to measure protein, we can do something called immunohistochemistry, or IHC, where we actually go on a microscope and look inside of the brain with these fluorescent proteins and see if the microglia are doing what we think they're doing. And we can use this data to make predictions about what proteins to go look for. So IBO1, as Brian showed you earlier, really important microglia protein. In these plots down here, blue means low levels, red means high levels. And so we look at this plot, well, IBO1 should kind of be in all the microglia. We should see high levels of it everywhere. GPNMB, on the other hand, well, there's only a little patch of cells that are high in GPNMB, and that little patch of cells lines up with part of this injury response. And so the prediction is, if we see a big injury response at 24 hours, within that injury, we should see this GPNMB protein. We shouldn't see it anywhere else. Okay, so that's the test. That's what we're going to go test. Oops. And we know where to look for it, as Dr. Cummings showed you. We know when we hit them 5 times, we get a thinning of the corpus callosum, we get some damage up here to the cortex. And so if we go and look in this area, we should be able to find some of this protein only in our injured mice. So let's test it. This is a healthy mouse. What we see up here in gray are your neurons. They're nice, bright, dense neurons, connections going all over the place. That's exactly what you want to see in a healthy brain. Injury response protein, GPNB, well, this is a healthy mouse. We don't expect to see any here, and sure enough, we don't see any red protein here. Nice and clear, okay? 24 hours after injury, we saw 60% increase. So 24 hours, we expect to see a lot of this red protein. Sure enough, we do. Not only are the neurons really disrupted, we see these big dark areas that we don't see here, but within those big dark injured areas, we see a bunch of this GPNB, this injury response protein, showing that the microglia are actively responding to that injury, just like we predicted with our colorful dots. But we had one more prediction we can make, right? We saw that it came up at 24 hours, and by 12 weeks, it went back down. And so if that prediction is true, if we look at 12 weeks in the brain, that protein should go away. And sure enough, that's what we see. The neurons actually seem to heal quite a bit. They look a lot better than they did at 24 hours. And this red GPNB injury response protein almost completely goes away. So at 24 hours, they responded, and then they went back down and mellowed out. And that was exciting because, hey, it looks like, okay, not only can the brain heal, but our data is reliable, which is good because we spent a year and a bunch of money on it. So we want to know it's reliable data. But it also, as we looked at these images, kind of highlighted another problem. And if we look at this little area and we zoom in on it, that slide. What do I keep clicking? I keep going the wrong direction.
Sure, clicking.
Oh, it's skipping a slide, I think. Okay, that's fine. Here, we zoomed in on this area. And what you can see is in the sham as we zoomed in on that little area, right? It's nice and bright here with the neurons. That gray in the background is nice and bright. During the injured 24 hours after injury, there's a big dark spot, and we have a lot of that injury response protein. And by 12 weeks, if you remember that zoomed out picture, the neurons look pretty good. But there's actually still a big hole in the brain left at 12 weeks. And so while a lot of the microglia inflammation came back down, and a lot of the response seemed to mellow out, there was what appears to be some permanent damage left behind in the brain. Those microglia couldn't fix everything. They went in and they tried and they cleaned it up, and a lot of the brain got better, but they left behind a little bit of damage. And that was 5 hits over 10 days. Imagine someone in the military firing thousands of mortars, someone playing football for 30 years, someone that's been in 50 different MMA fights, boxing matches. They're taking thousands of hits over decades. You're going to have these little bits of damage all over their brains. And so that is inevitably going to lead to disease. And that's what we're hoping to learn more about. So this is kind of the point where we're at right now. And our next steps are trying to figure out, is there some way we can address that? Can we improve upon that? Because these little bits of damage that are left behind are potentially long-term causing problems for people. And the way we're going to do that is, first and foremost, we still have a lot to learn. This is half the data that I showed you. These were just the male microglia. We also studied female microglia. So we have twice as much data as what I showed you. And I've been parsing through it, but I'm not nearly done going through it all and learning everything we can about it. We've learned so much things about pathways. We have a bunch of other genes. We showed you 2 genes. There's thousands of genes in here I need to go check. We also showed that human in these microglia, male and female microglia are actually really, really similar. And so this R squared value doesn't mean a whole lot to most people here. But if this was 1 instead of 0.92, it would mean males and females were perfectly correlated. They were exactly the same. So 0.92 is pretty darn similar between males and females, which makes us pretty confident that what we're measuring is really fundamental biology. It's something that's going to make sense for everyone, not just, oh, we picked this random cell line and so it's only relevant to the cell line. It's something that we think is truly human biology. We can also use some of this information to learn about biomarkers. There aren't good ways right now to track how badly someone's hurt. They get a concussion, and honestly, a lot of guesswork goes into it. Are they better? Can they go back on active duty? Can they go back and play? You wait a couple weeks. Do you feel okay? We'd like to actually know for sure. And so these microglia actually release proteins. There's some proteins they make and that they release. And our idea is that we might be able to measure some of those in the blood. And so if we look for some of these proteins after we hit these mice on the head in the blood, I don't know what I'm pushing, though. I apologize. Then we should be able to see that when they're hurt, those levels go up. And once those levels come back down, those mice are recovered. They can now, and so like say in someone in football, that would be the safe time for them to go back and play. And then finally, this isn't TBI. This is a recent paper that was published out of the Blurt Jones lab. But the idea here is that we can genetically engineer these microglia. So using things like Taq polymerase and PCR, we learned how to go in and amplify DNA and cut things and change genes and add extra little tools in there. And in this study, we took these microglia and we gave them an extra tool that helps them break down a protein that's associated with Alzheimer's disease. And they actually did it really, really, really well. So when we put these engineered microglia into these Alzheimer's mouse brains, they significantly reduced the amount of that Alzheimer's protein in the brain. And so we're pretty certain we can do something similar in traumatic brain injury. So we're going to use this CRISPR-Cas9 gene editing. We're going to edit these microglia and we're going to give them a new tool. We're going to take one of those response genes and we're going to stick a protective protein on the end of it. So again, going back to the idea of a recipe, let's say there's a recipe for bundt cake. And I go in and I staple on a recipe for ice cream. To say, hey, when you have bundt cake, you want some ice cream with it. That's what we're going to do here. And so now whenever these microglia go into this injury site, and they would normally turn on this gene, they'd make that bundt cake. After I engineer them, they'll also make this protective protein. So wherever you get this injury response gene, you'll also get some of that protective protein that'll hopefully protect those neurons. And the idea there is if you take a healthy brain, we injure them, we get this injury response. We're not going to be able to stop that injury response. There's already damage there that needs to be dealt with. Normally, that would lead to a bit of a improvement, but still some permanent damage. But once we add this protective protein in, we're hoping that we'll end up with a brain that looks a lot more like this healthy brain instead of having these big dark spots spread all over it. We got probably about a year and a half or so before we know if this works. This is the cutting edge of right where we're at today. Tomorrow, we're going to go back to work on this, and hopefully we'll get there over the next year and a half. And with that, these are a lot of people that did the work. I get to stand up here and talk about it and get all the applause and everything, but these are the actual people who did the work across a couple of labs, CERM funding, NIH funding. It's a team effort. It's a lot of us working together to get these things done. And thank you to all of you for being here as well. And then Brian wanted me to put up the rest of the seminar series. This is what's coming up. Do you have more you want to say about that?
Well, let's take questions from the audience first.
Yes. Thank you all.
Us.
When you do a concussion, you're hitting the midline, right? If you did it on one side, does it spread? Yes.
Good question. You want to repeat?
You want to repeat? Yeah. So the question for everyone on Zoom is, we're typically hitting the midline of the head when we do the injuries. And so the idea is if we hit one side rather than the midline, do we see that pathology, those injuries spread from one side of the brain to the other? Do you want to answer or you want me to answer?
Well, you go ahead.
I don't, have you done that directly and actually focused on one side specifically?
No.
No. Okay. So it's a good question. We do actually see multifocal injuries because they're on that pad. So the head does rotate and move. And so we will see, despite hitting on the midline, we'll see injuries in other parts of the brain. We'll see injuries on the ventral surface of the brain. We'll see injuries in the cerebellum because the brain, just like that video Brian showed, when you hit it and it wiggles back and forth, we get that in our mouse. And so it bounces and moves. So even if you hit one side, I don't know that it would be a clean single injury on one side. But I imagine there would be, because of that white matter damage, there would be some sort of disconnect that would spread from one hemisphere to the other.
Yeah, go ahead.
Any anti-inflammatories that might work or they don't?
Some folks have shown anti-inflammatories can help with concussion. We've tried some nanoparticle work that are anti-inflammatory nanoparticles that maybe we can deliver across the blood-brain barrier. But we have not shown efficacy with that yet.
Like part of the problem there too is like inflammation isn't necessarily bad. And so like a sledgehammer that's going to shut down, and you want some of that early inflammation because they're going to go in there and they're going to clean up that mess. And if we tamp all of that down, you may not get that same cleaning effect. There are benefits to early inflammation. It's the long-term inflammation that's problematic. And so you need to be able to sort of tease those two things apart rather than just sort of brute suppression of inflammation. My opinion, at least. Yeah.
So maybe this is a continuation of what we just said, like we see that the microglia correlate with injury, but we don't really know if they're good guys or bad guys.
Yeah. Yeah. And as again, for people on Zoom, the idea is, we see that they're in the injury site, these microglia, but we don't actually know if they're helping or hurting, right? We learned a little bit about the jobs they're doing, but are they really the good guys or the bad guys? And it's a great point. We know that they clean up. We know they go in there and they start clearing debris. We don't know how hard they work to actually fix things. Maybe it really is just as much as like, oh, there's damage. We're going to clear that out. And now we're inflamed and angry. And so we're going to make things worse. We were hoping to learn more about that long-term inflammation. So our data didn't show that they stayed really highly inflamed for a long period of time. Had we aged them longer, a year or so, maybe we would have seen some of that inflammation. I think there is damaging long-term inflammation where they do become the bad guys in a sense. I think early on, there seems to be a consensus, even in the Alzheimer's response, which looks really similar to this TBI response. They are trying to be good. And like evolutionarily, it sort of makes sense. If it was something that was right off the bat bad for you and you hit your head 1,000 years ago, well, you probably would have just died. So I think the evolutionary aspect is there is a good benefit to them coming in and cleaning up the mess, but we didn't live long enough for them the microglia to need to be good guys forever. So it didn't matter. If they became inflamed when you were 60 years old and you only lived to 50, it was irrelevant. Now we live to 80 years old. So that inflammation that comes up when you're in your 40s, 50s, 60s, all of a sudden that starts causing big problems in later age. And so they're a little bit of both, I think. And we don't fully understand what are exactly the good parts and bad parts.
Can you briefly describe how you're able to visualize these microglia reacting to the site injury?
Yeah, and the videos that we took. Yeah, so the question was if we can describe how we got those videos that I showed at the beginning of the talk. That was actually done in Sunil Gandhi's lab. And so it's something called a 2-photon microscope. And so a former graduate student of his, Dario Figueroa, he had a method where you could go in and thin the skull out. So they didn't fully cut a hole. They thinned it out really, really thin to where when you put that microscope right up against it, you could see into the brain, just on the surface of the brain. So it was right on the surface of the brain. Those microglia in that video were actually human microglia that had GFP, that green fluorescent protein in them. And so they were naturally friend. They were shining a laser through that microscope that caused that GFP to light up, and then they could record those green microglia through that little window in the skull. And what 2-photon lets you do is record over time, so you can get videos instead of still images.
That wasn't natural. That was Frankenstein.
It was, yeah.
Green microglia.
I copied that from a natural mouse microglia study done by first author was Davalos back in 2005. And so it was done in mouse with mouse microglia, and then I just kind of made the Frankenstein version of it for the sake of my study. Yeah. Yep.
Great talk. Did you look at the morphology of the microglia in a non-reactive or before injury and then after injury state?
Yeah, the question for people on Zoom is differences essentially in the morphology or the shape of the microglia before and after injury. We haven't done the quantification on it yet, but basically what we notice right off the bat is they become more amoeboid. They look like they're those activated phagocytic. Instead of having those long arms where they're surveilling, they pull those arms in, they migrate to where they need to, and they start eating up all that debris. And so again, we haven't actually quantified it, but that's broadly what it looks like with the injury site, similar again to the Alzheimer's response or any of the sort of activated microglia responses.
In the sham RNA-seq data you showed, it looked like that green population was there a little bit there as well. Do you have any speculations as to what that population would be doing in uninjured types?
Yep. So let me get to the next one. You make a great point. I actually simplified this plot a little bit for the sake of the talk. So the green population here is actually two populations that are kind of similar, but not entirely similar. And so the real like pure injury response is this little arm over here. That really is mostly down here in the injury and really not up here. Next to it is sort of a very similar state that has and that goes up significantly as well, but it seems like almost a pre-state or a primed state. And so this doesn't, you can actually see if I go forward to here, the GPNB really is mostly out here in this arm. This seems like some kind of way, I'm assuming it's a little more white matter related, where they are actively interacting with a lot of the lipids and a lot of things you would normally see in an injury site, but just naturally within the brain. They're in there doing these natural just cleanup tasks within the white matter that make them look similar to this injury. And then when we injure, we get this whole new branch that's really severely responding to injury.
All right, any last questions? All right, join me in thanking Johnny for giving a great talk. All right, and thank you all for coming.